Hello everyone, this video is about my research entitled Properties of Sourdough Starters for Bread Making and allows you one and the metric number is 187257. Some may wonder what is sourdough. It is a dough which is sour. It was made of flour and water which supplies sugar for lactic acid fermentation. Then lactic acid bacteria generates organic acids and carbon dioxide which makes the dough sour. Sourdough is used for bread making because dough properties, taste and flavor, nutrients, as well as shelf life are enhanced. But there are some problems. The awareness of sourdough and its benefits is still low in Malaysia. Then the local consumers are also not ready yet for bread which is sour. Besides, researchers for sourdough starters are still not acclimatized to our region. Therefore, we have some objectives for this research: to identify beneficial bacteria in sourdough starters, to compare sourdough starters from different types of flour, and to measure and predict physical-chemical properties of sourdough starters. Sourdough starters used for the study were wheat, rye, as well as sorghum. Why then? Wheat is used because it is the most common flour, rye is the most stable sourdough starter, and sorghum is gluten-free. In the research, 50 grams of flour was mixed well with 50 grams of distilled water. The dough was then left to be fermented at room temperature. Every day, the samples will be extracted for the tests like leavening height, the pH and TTA, the soluble sugar content. After this daily analysis, refreshing was done by discarding 100 grams of them and adding back of 50 grams of flour and 50 grams of water. The fermentation was done for 10 days and microbial test was done. For results and discussions, the first property measured will be pH. The pH of all sourdough starters decreased with time, as you can see from the graph. The lowest pH was achieved by wheat sourdough starter at a value of 3.26, followed by sorghum at 3.36 and rye at 3.86. The typical values of sourdough starters will be around 4, and the acceptable values of bread will be between 4.75 and 5. That's why manufacturers have to adjust the proportion of sourdough starter in the recipe of sourdough bread to ensure that the final pH of sourdough bread will not exceed 5, so that the sour taste will not be too strong for the customer. This table consists of the statistical models generated by best fitting the equation to the experimental data, whereby A, B, C will be the constants of this general formula: y equals to ax squared plus bx plus c, whereby y is the pH and x is the fermentation type. This general formula is used to simplify the determination of pH at a certain time. Like for example, maybe I want to have the pH of 4, so I will substitute 4 as the pH into y, and so that I can get which day will be the day that I can end my fermentation. And as for TTA, total tetratable acidity, it increased with time, as shown in this graph. The highest amount of TTA will be 14.8, achieved by wheat sourdough starter. The recommended maximum TTA for bread will be 6 ml. We have to put attention to avoid too strong sour and tangy taste in the bread. As for this general formula, y will be changed to TTA, and this is the table. And next, we'll discuss about the water-soluble free sugars. All of the sugars are peaked on day 2 for wheat at these values. Then sucrose is almost disappeared in all the sourdough starters because the presence of yeast in the taste make it hydrolyze quickly, and the maltose and glucose will have a second peak on day 6. As for fructose, it also increases due to its function as electron acceptor. As you can see from all the graphs, all the sugars will have low values at the end, towards the end of the fermentation. That's why the addition of sourdough into bread has many benefits. Like for example, it can have reduced surge of plasma glucose and insulin when it's being consumed. It is also healthy because it has slower digestion rate, which means you will not get hungry as fast as when you take a piece of usual bread. And all these changes of sugar in sourdough starters are due to the phases of microbial succession, whereby day 2 to 0, the microbes are sourced from water and cereal grains, and after that, some typical microbes will be lactobacillus, beta-coccus, and varicella. As for the last 5 days, the sourdough starters are usually dominated by sourdough strains. For example, Lactobacillus fermentum and Lactobacillus plantarum. Next, these are the statistical models. Next, for the leavening height. So as you can see from this graph, during every day of the fermentation, the sourdough is managed to rise the sourdough, all the sourdough starters, which the high increment is expressed in percentage for this graph. On day 2, the highest high increment was achieved by wheat sourdough starter at 82.91%, and the rest of them, they have either no or less gluten, so high increment is lesser, because gluten level is important to sustain the carbon dioxide produced by the LAB to rise the height of the sourdough starter. Then for the microbial populations, this is the graph for at class level, and this is the graph at genus level. Wheat has the least microbial population. As you can see, at genus level there are only 2 distinctive genus, and at class there are only 3 distinctive class. So the highest abundance in wheat, sorghum, rye are all under the class of bacilli, which lactic acid bacteria will belong to. The highest abundance will be in the wheat, which is around 90.21. And then these 3 are important LAB found in sourdough starters. First is Lactobacillus brevis. It is tolerant to low pH, presence of sodium chloride and potassium sorbate. All these 3 are the normal conditions in sourdough bread. That's why it's suitable to produce sourdough bread, and it also has the properties of extending shelf life of sourdough bread. Next will be Lactobacillus paraplantarum. It is collectively known as probiotics together with Lactobacillus brevis, and with these 2 strains. Probiotics actually it means that they are living microbes which brings positive health effects towards the host when they are being administered. Some common example of food products having probiotics are like Yakult, like Vitagen, like all those yogurt or yogurt drinks. They will have this kind of probiotics which is good for the health of our guts. Besides that, Lactobacillus paraplantarum, they are antibacterial and antifungal also. As for the last one, Lactobacillus helvetica, they are potential probiotics too, because they have a lot of health benefits like this. In conclusion, sourdough is very beneficial as it gives distinctive taste to bread at suitable amount according to customer preference. It also has self-leavening properties, thus it has potential in replacing industrial yeast. Sourdough bread will have longer shelf life due to low pH and antimicrobial properties of LAB, hence less chemical additives are required. Through fermentation, LAB consumes soluble sugar, which then reduces sugar level in sourdough bread, making it healthier to be consumed. As fermentation goes, pH decrease, TTA increase, fermentable sugars changes with 3 phases of microbial succession, and the leavening height was maximum at day 2. The values of studied properties within fermentation period of sourdough starters can be estimated easily and rapidly by applying the statistical models generated. Wheat sourdough starter has the least microbial diversity, and the important LAB detected are as below. Thank you and have a nice day!